So hopefully I can add to this very rich conversation that we've had already today, and you'll see several, as we already have, common threads woven throughout a lot of the work that we're doing. I'm going to talk to you today about work that I've been doing in a number of longitudinal birth cohort studies to start to understand early life social and environmental influences on lifelong health. Starting out, I spent a lot of my time looking at risk. Now I'm really turning my attention to talk more about resilience and what can we understand about that and what types of interventions we can have. And it's interesting, I think, to step back and hear about how I got here, because I'm not a pediatrician that's in the Department of Pediatrics and a professor of children's health research. I'm actually an adult-trained pulmonary critical care physician in my medical training, and started out studying genetics. I spent years doing genetics research, starting out at the University of Michigan when I wandered onto the campus to start to study pre-med. It was going to be the genes that made us all healthier, they said, and I bought it hook, line, and sinker. My undergraduate research, or my undergraduate degree, I was the first person to graduate from the University of Michigan with a degree in human genetics. I petitioned the Honors College because I was so into it. I did all this research. I spent a year at NIH during medical school there. I spent my year between my second and third year. So from between the classroom and seeing patients, I thought I'm going to get a PhD in genetics. I went down and spent a year to decide that. Came back and started seeing patients, and I thought it is not going to be the genes, because now I was learning and seeing what was, and to Rachel's point, you got to listen to your patients, listen to your communities, and that's where it really took me in a whole different direction. And then as a practicing intensivist, pulmonary and critical care physician, taking care of end-stage disease, taking care of people in the ICU with multiple comorbidities. Even in the first 10 and 15 years of practice, I could see shifts in what was happening there, the obesity, the multiple comorbidities in patients. And I felt a little bit like this. Your person by the river hears someone drowning. So what do? Being a good swimmer, you rescue the person. You jump in, you can swim, you get them, you bring them up on the bank. Before catching your breath, though, there's another and another and another. And exhausted, you begin to walk away. And someone asks, where are you going? I'm going upstream to prevent others from falling in. That's what took me as an intensivist looking at people at the end stage of their disease and to say, how far back do I need to go to really move the needle, put people on that different trajectory, like you heard about in the last presentation. And boy, I kept going back further and further and further, and it's really here where the roots of it is back here. So now I had to learn to become not just a genetics researcher, a clinician, but now I had to learn environmental medicine. It was really environmental factors. Then social determinants became an issue, so now I had to learn that and stress. This is why I call myself the transdisciplinary life course. I don't know what to call myself anymore, so that's what it is today. And this, this hit the Time magazine the first 9 months. It's not over, as you heard before, but boy, is it a place where we really can have big bang for our buck if we can understand it and start to understand what those factors are. We really measure all of these different components. You've heard about the multiple components that drive health, and they're certainly important at the roots of it, and that includes nutrition, chemical exposures, pollutants, psychosocial factors. When I think about that, I think about stress as how they get into the body, and built environment. We do it all, and I'll tell you, the one that rises to the top for me, and I'm so glad I listened to my patients and had my eyes open to understand what they were showing me and telling me, because we measure all those things. It really is the social factors that we need to get our head around if we're really going to move the needle on this. So things like violence, poverty, we've heard about all these things today. I think about them as social pollutants. Just as you breathe in toxins and tobacco smoke and allergens, these things get in, and they imprint all the way down to the fundamental levels of our genetics. This isn't a new idea. There's a book, Raising Children in Socially Toxic Environments, written back in the 70s by a psychologist that's worth the read. Social toxins are as detrimental, if not more so. That's what I think I've learned from some of the work that I'm going to show you today, than these other physical and chemical toxins. And moreover, they enhance vulnerability to these other chemical toxins. Even at lower doses, they're going to be toxic if we don't pay attention to the social factors and the stress underlying that. And they begin prenatally, and they're transgenerational. They're carried across generations, and that is daunting to realize and understand. But it also speaks to maybe how we have to think about how we craft our interventions and who we need to intervene upon. So really, the big motivation for me was in really shaping this work, going from genetics to all of this, was really to try to understand and tease apart socioeconomic and racial disparities in lung disease. I was trained as a pulmonologist, so that made a lot of sense. Asthma is starts early in life. It made a lot of sense to do so. But I do measure all the, you could, put in neurodevelopment, obesity, whatever you want for the outcome. It means the same thing. And what I'm trying to understand is, can we measure life stressors? Can we measure how it gets in the body? And can we convince ourselves it's involved in health, growth of the respiratory system and lung health and disease, starting even in those early periods? You saw earlier talks this morning that gave you an excellent overview of the stress pathways that might be involved. When you experience stressors, psychosocial stressors, it can disrupt these key regulatory systems that are in your body to help maintain some optimal balance and keep us all healthy. When those things get pushed and pulled and shifted from chronic stress, that's, in the short term, that's adaptive, but in the long term, there's a health cost, and that's what we're trying to get our heads around. And we look at not only the HPA axis that we heard about, cortisol, the hypothalamic pituitary adrenal axis, but the autonomic nervous system imbalance, oxidative stress imbalance, as well as these things talking back and forth to the immune system. So when you think about asthma, it's a disease of disrupted immunity, makes a lot of sense. Now, when that happens, when the mother carries in disruption of those key regulatory systems in pregnancy, it already starts to shape development in the child, development of their own stress regulatory systems, their own ability to self-regulate and improve, and respond to the environment in an optimal way, and also disrupts their immune system and the organ systems that are developing in that context, and certainly the lungs are going to be important there. Just as the things that you see in the blue box there, microbes, smoke, air pollution can disrupt these systems, so too can psychological stress. And they can interact. And I'm going to focus on one chemical pollutant in particular. I'm going to focus on air pollution to just exemplify some of that, but we look at some of these others as well, and you could, again, substitute any of them. So how did we go about deciding how can we measure this? Well, as a pulmonary fellow at Harvard, I went around and said, what cohorts are available that I could ask some of these questions in, because I need to have measured all of these different things, not just the stress, but I'd like to have rigorously gone after things like the air pollution, allergens, diet, to nutrition, and whether or not these folks were exposed to tobacco smoke exposure during this time. Problem was, no one had really measured stress at all at that time that I was looking around in any of these cohorts, and let alone had concomitantly looked at these other things. Why is it important? It's important because at the end of the day, somebody was going to say, if I just measured stress, it's just a surrogate marker of bad environment. And I heard it, so I knew it was going to be said. It was said by my colleagues, so I said, okay, I got to do that. So we developed, and we're lucky enough to get funded to do the Asthma Coalition on Community Environment and Social Stress. This was a community engaged project where they also weighed in on what the important things that we might want to measure would be. And this just shows you a timeline of lung development, just to exemplify that the periods of critical windows of development for the lung starts very early. If you look at the your left-hand side of this slide, I'm not going to point because I know people on the WebEx can't see the pointer, so I'll try to describe. Look how rapidly the growth of the lung and the tubes and all of this has to happen over the course of prenatal environment. But it's not done there. There's also rapid growth that continues in particularly those first 2 to 5 years of life. And the lung is an interesting organ system because it continues to grow all the way to late adolescence, early adulthood. So there's vulnerable periods throughout, but the most plasticity is that early period, just as we heard about before. So again, that's going to be important. And in the timing, it's so intricately orchestrated, the development of the lung during the prenatal period, that we've been very interested in developing ways to measure exposure across the whole gestational period and use data-driven measures to point us to where those windows of vulnerability are, because maybe then we'll understand mechanism or probe mechanism better and hopefully inform interventions, timing of interventions better. So when we went about measuring stress, some of the things we learned, and we've already heard about it with the other talks, the nature of the stressor can have different impacts. The timing of when that happens, either in the mother's life or in the child's life, is important to the outcome that you're looking at. And boy, are there gender differences, and that's something that's been ignored, but we're really starting to see that. So with that preamble, let me tell you some of the data that's come out of that Access project. We first looked at the independent impact of psychological stress. What do I mean by independent? It's even adjusting for all those other environmental factors that were floating around the little kid there in my graph, it's still significant, and also adjusting for socio-demographic factors. Now, we heard a little bit about how to measure stress and think about what's impactful. I just want you to think about this little guy running up over the hill. And when external challenges overwhelm one's ability to cope, so when you get up to the top of that hill, that's about all you can manage. And when they're starting to tip you over the other side, too much, too many stressors, too high impact, you don't have the coping skills or the social support networks or whatever it takes to help adjust and buffer those. That's where you start falling off, and that's where you start to see the potential physiologic effects that will translate into adverse health. So one of the ways we measured it and thought about it was if you're experiencing too many at the same time, too many challenges at the same time, that's when I fall apart, right? I can manage a lot of things, but it's when all of a sudden, too many things are going. I have to say to myself, something's got to give. I got to bring that back into a new balance. So we used life event measures that measured across a number of domains, and we use this measure called the Crisis and Family Systems measure, which measures across multiple domains, some of which are shown here. Experiencing financial strain, racism, discrimination, relationship issues, community, interpersonal violence, not only at the individual level, but also at the community level, things that you're experiencing. And other negative life events, and housing was a big one. That was one that our community was telling us about. So housing is often overlooked in some of these studies, but that's certainly part of it. And so what we did is we looked at a sum of those life domains that you're experiencing and tried to see, is there an association? And what we found is shown here. On the left-hand side, it's that measured prenatally in our Access women during pregnancy. How many of these life domains are you experiencing stressors in? And in the right-hand panel, it's the postnatal in the first 2 years of life. So those two critical windows. And I had to send my postdoc out the door and say, look at that again, because there's no way that's as dose response as I'm seeing there. But it is, and we see it over and over and over again. And that's very convincing to me when you start to see a dose response effect in association with here. In this case, we were looking at an early marker of childhood asthma, which was repeated episodes of wheeze, having 2 or more episodes of wheeze in the first 3 years of life. Again, adjusted for all those other things, those other environmental factors that we know contribute to that. We've also since looked at that in another cohort. This is a Mexico City cohort that was designed in parallel with some of the same measures that we used in our Boston cohort. And I show it because here we recently published in the Annals of Allergy, Asthma and Immunology these gender differences and when the stress has an effect. And on the left-hand side of that graph is everybody lumped together, and it's looking at prenatal stress on the, whoops, lost you. And it's looking at prenatal stress on the top and postnatal stress on the bottom. And the boys are in the middle panel, girls are in the right-hand panel. And look at the difference. The boys just have that dose-response cumulative increase in effect with increasing stress in their mothers during pregnancy and whether or not they had repeated wheeze in early life. Girls are flat during the prenatal period. Now look postnatally. the girls may be protected prenatally, but they have at the cost post, if the exposure continues, they are even more vulnerable. And other studies have shown this in animal models, et cetera. It parallels what's seen in animal models. This was very impressive to me. The other thing that we know is that if you have cumulative stress, like if moms experience that stress, high levels of the life domain stressors, both in the prenatal and the postnatal period, their kids are most at risk. So here we're showing on the left-hand panel moms who reported were scoring low, both pre and postnatally as the referent group. And if they had high only in the pre, is the next group high only in the post? Yeah, there's some increased risk, but doesn't even quite reach statistical significance. But boy, if the moms of these infants have cumulative stress in both periods, they're the kids that are going to be at particular risk. We were doing this to try to say, well, should we intervene pre or should we intervene post? The point is, you can start it. Either of those periods is fine, but probably we should start prenatally so that we can give the kid the best chance, right? Moms and kids. 6 years later, we can measure lung function in these kids. And what we found is that there's also a cumulative relationship with levels of lung function in these 6-year-olds. So we brought them in, we had them do spirometry, and you can see again, we looked at the cumulative high in those two periods, those two critical windows, prenatally and postnatally. And the ones with the lowest lung functions are the kids born to mothers who had high levels of stress in both periods. Again, it's adjusting for all those other environmental factors. So again, it's independent, individual effects. Now, why is this so striking? Because your level of lung function at age 6 or 7 tracks. It tracks over your life course. You've already reduced the maximal level of attained lung function that these kids are going to see. And that puts them at a huge disadvantage. They're going to be more vulnerable to subsequent toxins. They're going to be more at risk for developing chronic obstructive lung disease and all comorbidities that go along with that, and even early mortality is linked to levels of maximal attained lung function in life. So again, it might be costly to intervene down here, but if you're looking at a lifetime of what that brings for this kid, it may be where we need to put our resources. So early life toxic stress is literally taking our breath away. It's very important. It's those early life periods. Stress is a toxin, just like these other things. We can't ignore that, and we need to think about how to put that into the conversation with all these other things that we have to deal with for sure. The other thing about timing of exposure, we were talking about mom's stress during pregnancy in the last 6 months. Which of these life stressors have you experienced? I don't think I made that point clear before, but I'd like to now, because it's not just what happens to that mom during pregnancy that we have to be concerned about. It's what her life course has been up until now that actually has an impact, and what she carries into that pregnancy. And it might be something that happened to mom in her own childhood that's the most impactful. And we've seen this in different ways. So we also looked at specific types of stressors over the mom's life course, and trauma is a unique stressor. It's the one that gets imprinted. This is a Gary Larson cartoon. All the forest animals sitting around saying, "Where were you when Bambi's mother was shot?" Not a human experience that any of us should have, and it's a stressor that has impact. It's going to have a lightning bolt on these systems. It's going to reset these systems, and it could carry on for long term. So we've measured cumulative traumatic stressors in our mothers. We've asked them to talk to us about interpersonal trauma over their life course. And we asked it in a way, we asked, over your first 0 to 10 years, did you experience any of these things with a standardized measure? 11 to 17. 18 to the pregnancy and during the pregnancy. So we asked the same questions over 4 life periods, and we looked at the number of life periods that mom experienced some kind of traumatic interpersonal trauma. And we related it to child health outcomes, right, in the next generation. Here we looked at, and we published on maternal interpersonal trauma over her life and cord blood IgE levels. So elevated IgE levels in cord blood, not a perfect marker of who might go on to develop allergy and asthma, but boy, if you're high on that, you're going to be likely to if your environment sort of pushes you in that direction after the birth. And you can see that compared to never in the left-hand panel, the levels of IgE there demonstrated, to 1 to 2 life periods to 3 or more life period she's experiencing these types of events in her life, child's more likely to have their immune system pushed towards an allergic type phenotype. Here we looked at maternal interpersonal trauma and what the pathway was. Don't get caught up in the figure. What I want to show you is the bottom path was the important one. It was mom's own health. So if mom had interpersonal trauma, chronic interpersonal trauma, she was more likely to have active asthma during that pregnancy. Her asthma was more likely to be active, and that is the pathway to her child being likely to be an asthmatic. And there's data to show if mom's asthmatic, that whole immune milieu that the kid's developing in is very different, and they're going to be at risk not only for asthma, but for neurocognitive problems, et cetera, et cetera, et cetera. Isn't it interesting? So it's the mom's health, too, that we need. That's another pathway that we need to be concerned about. We did look at community-level stressors, and I just want to show on the left-hand panel was increasing community violence exposure in the communities that they grew up, the moms were experiencing during their pregnancy. And again, you see a dose-response effect. So moms who were pregnant with the highest level of violence, their kids were more likely to go on to have wheezing, respiratory illnesses, and asthma, and everything else that we looked at. The reason I show the right is that's air pollution measured at the mom's residence during pregnancy. Same dose effect. It's the same magnitude of effect. Both of them are independent predictors, but the same magnitude of effect. So it has the similar magnitude. This is just a meta-analysis of studies looking at prenatal stress, predicting respiratory outcomes, and a meta-analysis showed that overwhelmingly, if you're on the right-hand side of that bar, that means there's increased risk. So studies have consistently shown, it's not just the work that I'm showing here. Some of the work we've done is on mechanisms. So we've looked at imbalance in immune function and showed that increasing stress, which is on the axis on the bottom, as you go across there, you have immune changes that are very consistent with being at greater risk for developing asthma. So it does seem to be affecting these key systems that we're talking about. Here we showed that cortisol levels were elevated in the evening time. So that, and someone talked about that in the earlier talk, that was a risk factor. And these were the ones, moms who had high cortisol in the afternoon and evening, their kids were more likely to develop respiratory disease postnatally. What about interactions? We also looked at air pollution across pregnancy. So gestation is on the bottom axis from 0 to 40 weeks. And we had measurements of traffic-related air pollution, particulate matter, measured for the women, averaged over every week of pregnancy. And we used a method, a statistical model that could tell us where the risk was, what was the window of vulnerability. And we found that in mid-pregnancy, in the second trimester, is when air pollution seemed to have the greatest impact on risk for asthma. But boys, if you looked at the left-hand side, what we're telling you is boys are at greater risk than girls. Again, we see that with air pollution just as we did with stress. Now let's look at double jeopardy, air pollution and stress. And it's really in the high stress group only that we saw that relationship with air pollution. That was a significant interaction. And then now a triple jeopardy, right? Chemical vulnerability, social vulnerability, and then biological vulnerability, in this case, gender. It's only in boys born to mothers with both high stress and high pollution. And again, that's a significant three-way interaction. So what about resilience? What are the things that we're starting to think about and look at? During early childhood, we've heard nurturance from the caregiver is a big driver of health in kids, and it's also the big driver of developing their own ability to develop resilience to these things. We did a study looking at moms, kids born to moms on Medicaid, and they had measured domestic violence in the home at the time of pregnancy, first 12 months of follow-up, and at 3 years, and they also measured who went on to get asthma. And we showed that there was a twofold increase in asthma in kids growing up in domestic violence households, but there was also a measure of supported caregiving. How often do you sing to your child? How often do you read books to your child? These kinds of things. We heard about that a little bit. It mitigated the effect. That got me very interested, and we developed another study looking at follow-up to the Access Project, similar measures of risk, but we also started looking at programming of intergenerational stress mechanisms in these kids. And we brought them in the same design as I just talked about with all those risk factors to the prenatal exposure. We brought them into the lab at 6 months, And we actually measured caregiving sensitivity by observing through a videotape and coding very intricately the caregiving behaviors. And we actually looked at that on the effect of the negative affective response, HPA axis, and autonomic functioning of these kids. That's just the stressor that we used. And what I want to show you is this. The dotted line is the insensitive caregiving. And over the course of that stressor, the kids where the mothers were coded as not insensitive, they had much greater heart rate response, much greater distress, and were less able to recover from the stress challenge. And this was over just a 6-minute lab thing. We also measured cortisol, and this was even more remarkable to me. Again, the dotted line is the insensitive caregivers. The cortisol went much higher, and they took much longer to recover. So already there's suggestion that their stress response systems are not operating in an optimal way. Insensitive caregiving seems to be something that can actually mitigate the effects there. So we want to put that in the context of, does that then decrease your likelihood of going on to develop asthma, obesity, et cetera, et cetera. I want to just take 1 minute to say something about nutrition, which are the slides that I added, because it is so important, and we're starting to look at it as well. So we measure maternal nutrition in our study in prenatal period using a food frequency questionnaire validated with 24-hour recalls, because it's a very ethnically mixed population, so we thought we needed to do that. And there's data to show that polyunsaturated fatty acid intake and vitamin E are factors that can ameliorate the effects of certain oxidant toxins, like air pollution, like tobacco, but also stress is a pro-oxidant toxin. So could it mitigate the effects of that? And the way it would work is that the increased oxidative stress induced in the placenta, transmitted to the child, and effects on the developing brain could be mitigated if we looked at moms who had high intakes of polyunsaturated fatty acids and or the antioxidants. We measured that in our kids looking at a measure of negative affectivity through a questionnaire at 30 months of age. And this just shows you. So these 4 blocks are just antioxidants: vitamin A, vitamin C, zinc, and selenium. The high antioxidant intake is the dotted line. And so if you look at increasing stress, prenatal negative life events over the bottom axis, as you go from left to right, it goes from low to high. And you can see the effect on negative affectivity in the child is flat with all the dotted lines, the high intake of antioxidants, but it goes up, and that's where you see the detrimental effect when the mom had low levels of intakes of antioxidants. So that's some of the first data to really show that maternal prenatal diet can mitigate the effects of that. So this is just the figure you got to see. As an internist, a critical care physician, I was out here at the right-hand side of this graph trying to move the needle, make people's health better. Where's the bang for your buck? It's all at the beginning where the plasticity is. And looking ahead, it's easier to build strong children than to repair broken men, which is what we're doing at the end. You know, we can do things for sure, but we can't move it that much. So thanks very much. And just to say, I want to applaud all of these folks who helped us do it.